In this lecture, I'm going to talk about some concepts that were not covered in the guest Q&A or the student mini lecture. I guess making this lecture kind of after all of that was done, I could, it's actually nice because I get to see, okay, what are some concepts that are important that weren't covered. So the first is weed competition with crops, resource versus non-resource competition. At the end of the day, weeds are bad, they're going to reduce yields, but there's two ways they can do that, and there's some implications, practical implications, but more theoretically, I think it's important to understand this. So the first way weeds reduce yields with crops is resource competition, which is just straight up, the weeds are taking the water, the nutrients, the light away from the crops, and that's bad. Then there's non-resource competition, which is pretty broad. It's basically any other mechanism by which weeds will reduce crop yields aside from resource competition. So anything else outside of that is non-resource competition. And I'll explain this in more detail. It's quite general, like what does that mean? But first I want to introduce the concept of the critical weed-free period. Now this is the window of crop development where weed competition is most strongly reduces yield. Just like we have a critical period of yield formation in corn, soybeans, and wheat that we talked about earlier in the course, there's a critical weed-free period where if weeds are present during this window, this time frame, yields are much more strongly reduced than if weeds are present after this time frame. And we'll talk about, I'll give you some more information on the critical weed-free period for different crops, but it's generally early in the season, okay, soon after emergence. Okay, so how does a farmer figure out, the critical weed-free period for a crop? Or I shouldn't say a farmer, it would be a scientist. Now, this concept of the critical weed-free period was discovered at the University of Guelph, largely by Clarence Swanton, a professor who retired recently, and it was adopted like industry-wide, like globally, this concept of a critical weed-free period. The implication for a farmer is you got to make sure weeds are controlled during this time. And so that's why in Ontario especially, there's a lot of emphasis on pre-emergent herbicides applied prior to planting, soon after planting, to keep weeds out of that field during the critical weed-free period. So how do you figure out what that critical weed-free period is? It's not that hard of an experiment to run. It's kind of interesting. Let me delete that. So you're going to take a field that's weedy, or maybe you'll plant winter wheat to act as kind of a surrogate weed. Let's say we want to know the critical weed-free period of quinoa, really delicious grain, high in protein, actually relative of spinach, very affordable too. So you're going to have a couple of treatments. You're going to lay these out in your plots. You're going to have your 100% weed-free control. So generally these experiments are done by armies of summer students that hand weed these plots to ensure like 100% control of weeds. So they're hand weeded like every day or every 2 days. So this would be a plot that is weeded every 2 days or so for the whole crop life cycle, okay? And let's say that's, 100 bushels per acre. Then you have plots that are 100% weed control. They're never weeded. They're just weeds are allowed to grow. Let's call that 40 bushels per acre. Then this is where it gets interesting. And this is how you figure out your critical weed-free period. You'll have a plot where you keep the plot weed-free for 7 days. Say, this should be after emergence, right? So you're going to hand weed that plot for 7 days. But then after 7 days, you're going to let the weeds run rampant for the rest of the season. They can just keep growing. Then you're going to do basically the same at different time intervals, right? You'll have weed-free for 14 days, weed-free for 21 days. So you'll be weeding for 14 or 21 days, but after that time period, you just let the weeds run rampant, okay? And let them keep growing. So let's think about what's happening in these treatments, right? You're keeping the plot weed-free for a certain period of time, and then you're letting weeds just keep going. What we see here, and this is a simplification, but it's basically what you do to determine this weed-free period. We notice that after 21 days, our yield, 95 bushels, is very close to our 100% weed-free control. Now, our yields are just 5 bushels off, despite the fact that the only difference between this treatment and this treatment is 21 days, right? Because this plot was kept weed-free for 21 days after emergence. Then the weeds could keep growing. So what this is telling you as a farmer or scientist, agronomist, is, wow, okay, doesn't matter if weeds are there 21 days after emergence. Right? You really got to make sure that they're not there for the first 3 weeks, 21 days of that quinoa life cycle. Right? That's where you really read to make sure you're not getting any weed interference. So this is kind of what this is actually from a study. This is This is data from a study that was done in Ontario to determine the critical weed-free period for quinoa. All right, so this is what that graph looks like. You have the relative crop yields here, so percent of weed-free control. So whatever yield was, it was set at 100% of the weed-free control, no weeds at all. And then we have yield, relative yield. If this is the 100% weedy control, so no weed control at all, and that was 50% of your weed-free control yield. If you kept quinoa weed-free for 7 days, you got 75% of your relative yield, so you only lost about 25% of your yield. If you kept the weed-free period 14 days, so you weeded every day for 14 days, but then let the weeds run rampant, you were essentially at the same yield, almost the same yield, as if you kept that plot weed-free the whole time from basically emergence. So what this is, and then they do some fancy statistics. It's not like as simple as I just explained it. But what this is saying is that just after 14 days, this might be 15 or 16 days, that is the critical weed-free period. If weeds emerge after 16 days and they're not controlled, they won't reduce quinoa yield. Okay, so the critical weed-free period has been determined for a number of Ontario field crops. This is from the OMAFRA Agronomy Guide. I'm just cutting and pasted here. Corn is the 3 to 10 leaf tip, which would be V2 to V8 using the leaf collar staging. So this is the critical weed-free period for corn, and then you have for everything else. A couple of other things for this critical weed-free period. It doesn't really vary by hybrid or cultivar, but it does change a little bit with management. So a whole lot of things. Narrower row spacings typically reduce or shorten the critical weed-free period. Basically, anything that kind of makes the crop less competitive with weeds is going to lengthen that critical weed-free period. There's also the effect of planting date and weather here. And this is just wanted to highlight this. This is an Ontario study looking at the critical weed-free period in carrots. All right, so this is carrots, and so we have 5 locations in different years, 2007, 2008. They basically did the same kind of study to determine the critical weed-free period. And here they said critical weed-free period is where you get 5% yield loss or less. So in quinoa, it was like around 15, 16 days. Generally, you'll standardize that to growing degree days just to make it easily comparable across location years. So growing degree days here. Notice that it's a pretty big difference in the length of the critical weed-free period. You have 3 location years where it's very close together, like the 400 to 500 something degree days, or the 4 to 6 carrot leaf number stage. But then you have these 2 that are the critical weed-free period is much longer, right? To 10 to 12 carrot leaf number, and correspondingly a lot more growing degree days. So, what's going on? Like, what could be causing this? The study was not super replicated. They didn't keep things like row spacing. Planting date was a big difference between these sites. I'm not sure the cultivars might have been different, although that probably would not have had an effect. I don't know. I've read the paper. The paper isn't sure why, but I was interested to see, wow, it can actually be quite big in terms of how variable this critical weed-free period is. So I would take these as really good guides for the critical weed-free period, but it does depend on management, possibly weather. All right, so let's bring this back to resource versus non-resource competition with weeds. So basically what the weed-free critical period is telling you is that, after the end of this time, the crop is large enough, more developed enough that it can compete with weeds successfully. So, for example, with quinoa, after 16 days after emergence, or in corn after V8, the crop is big enough that weeds that emerge are just going to be smothered out. They're not going to be able to compete with the corn. So basically, this is better resource competition. The crop has an advantage there because it's big enough. But this is something we see like in these studies, and I want to point your attention to it, that with quinoa, right, this is the yield reduction from keeping the plot weed free for 7 days or 14 days is huge, right? You go from 75% relative crop yield to basically almost 100% relative crop yield, right? So there's almost no yield reduction, actually, on average, almost no yield reduction here. So this would be something like 97%. So you're getting almost the same yield, keeping it weed free for 14 days. So there's a huge difference in yield just by keeping the plot weed free by an extra 7 days, right, from 7 to 14. We see this in other crops, not just quinoa. And the question is like, what is going on? Why is there such a big difference in yield between these two treatments? Think about it, right? From 7 to 14 days after emergence, doesn't matter what your crop you're talking about, nutrient uptake needs, water uptake needs, light needs are minimal, right? The crop does not, it's so small, that crop, it doesn't need a whole lot to keep growing. And so the chance that weeds are going to have enough resource competition, they're going to be stealing enough nutrients, enough water and light to reduce crop growth or eventually yields during the 7 to 14 day period. To me, that it doesn't make sense that this would be really a resource competition thing. What is going on here is non-resource competition. Basically, during that period from 7 to 14 days after emergence, the reason why yields are reduced is because that plant, those plants in the field are going to permanently redirect biomass, that is carbon, towards vegetative structures. Things like the stem to get taller, right? It's going to sense neighbors, weedy neighbors. It's going to say, oh my goodness, I'm surrounded by weeds. I'm surrounded by enemies. I need to grow really, really tall as fast as possible. And this switch in the mind frame, it's like almost like trauma. It's almost like permanent like PTSD that the crop goes through. Because even though it's experienced a couple weeks after emergence, this is going to carry through to crop yield because carbon is permanently going to go away from the grain towards vegetative structures, deeper roots, longer stems, and so forth, maybe more leaves depending on the crop. So this is what we call non-resource competition, and that's what's really driving this yield difference for a 7-day period right after emergence. Now I just want to give a few thoughts on plant diseases. There are fungal, bacterial, and viral diseases, and it's important to know what you're dealing with because if you spray a fungicide and it's a bacterial disease or a virus, you're not going to really do anything at all. So there's fungal, bacterial, viruses, and then you can further kind of figure out what specific disease is occurring by seeing what is being affected by the disease. Is it the seedling? Is it roots? Is it leaves? Is it the ear for corn? Or is it the stalk or stem issue? Often a disease will only attack a couple of these, not all of them at the same time. Some other thoughts, right? So spraying a fungicide or insecticide, timing is very important. So there's usually a window, let's say, I don't know, it depends on what's going on, but a week, maybe 2 weeks at most, where spraying is going to be effective. So you either need to scout very, very well or just have it part of your planned program where you know at this stage we're going to go and spray. Timing is very important. So too early or too late is going to really reduce the efficacy of whatever you're spraying. Another thing here, I wanted to bring this in because it seems like every single group in the in our discussion said plant a resistant variety or choose a resistant variety, and like absolutely yes. But and I know farmers do this, and it's important to think through. If a resistant variety or a tolerant variety, it's usually tolerant, not resistant, is lower yielding, then just you got to think through risk management. Often the hybrids, from what I've seen in corn, hybrids that have the biggest yield response to a fungicide application are normally susceptible, right? That makes sense, right? Because if you're tolerant, applying a fungicide is not necessarily going to have as much of a yield impact. And it's obviously a risk management, but I think I've known enough successful farmers that I've said, you know, some fields I'll plant tolerant hybrids, but because I really am chasing yield potential, I'm going to plant higher yielding susceptible cultivars or hybrids and plan a fungicide application. So just a different perspective. I'm not saying one's right or one's wrong, just something else to think about. And then the effect of climate change on diseases or pests. So a couple of points. Pests are always changing. Like regardless of climate change or not, there's just this evolutionary pressure. It's like an arms race between us, the farmer, agronomists, scientists, humanity, and the pests. Right? We're throwing something new at them, and they're going to try to build resistance and overcome whatever we're trying to throw at them. So it's just an arms race. So in terms of R&D, it seems like you're working harder and harder to stay just in the same place, right? Because you're doing all this R&D to overcome pest resistance. That's a lot of money, but it's not necessarily increasing yields. It's just stopping what could be massive yield reductions. And in Canada, it's pretty clear that climate change is introducing new pests. If you ever, in your future career, end up sitting on boards, like farm boards that basically review proposals, like Grain Farmers of Ontario has a scientific committee where the farmers will review proposals and whittle things down. A lot of proposals have to do with like new diseases and new pests that are coming up. Tar spot was the big one that was brought up in class, but there's a lot of other ones. And it's tricky because we'll see a new type of disease show up in Canada. It'll be in a couple of fields. And you don't know, is this going to be a huge problem and something we need to invest lots of money into right away? Or is it something that'll blow over? And actually, yeah, we saw it in a few fields, but 5 years later, it's amounted to nothing, right? That has happened in the past. Some new races of Fusarium head blight have come up, and we thought, uh-oh, like this could be really bad. And it turns out, actually, it's not as bad as worst case scenario. It's very hard to make those decisions because you don't know as a farmer and you're saying, should we fund this proposal or not? This is a new disease or a new pest. It's been found in 5 fields. Will it be found in 50 fields next year, or is it still just going to be 5 fields? Very hard to say. So it's very tricky to deal with climate change in terms of how to manage R&D investments. Okay, so next I want to talk a little bit about this is more for your own information. It's not really practical agronomic knowledge, but we often think very simplistically about like resistant versus non-resistant cultivars, right? Like, oh, this is a resistant hybrid. It must have a gene that gives it resistance. And that's just not true. So I'm going to give you some case studies of like what makes a resistant variety resistant. This is not like, at the end of the day, from a farmer perspective, doesn't matter, but it's interesting and I think you should know it so you actually understand like what makes something resistant or not. It's more complicated, more interesting than you think. So our first case study is based on a really nice paper. We won't read the whole thing or go through it, but a paper that figured out why wheat varieties that are tolerant or resistant to Fusarium head blight, like why they are resistant. So wheat breeders have been breeding for resistance or tolerance to Fusarium graminearum, the fungi species that causes Fusarium head blight, a big problem in some years, especially in winter wheat. And so they've been successful at it, but breeders have not known exactly like mechanistically what's the cause, like what is causing resistance in resistant cultivars. They just know they have just bred for resistance through basically just doing lots and lots of studies of field trials. Now, let me just back up a little bit in terms of, plants don't have an immune system. So how do they deal with fungal infection? Like what tools do they have if they don't have, like we do, an immune system? So there's a couple of things that they generally can do. First off, fungal diseases, especially necrotrophic ones, will, their mycelia will go across cell walls and they'll inject the plant cells with things like DON or different toxins, right? They're trying to kill those plant cells. And a lot of those toxins end up being poisonous to us or animals as well. So DON is secreted by Fusarium head blight, deoxyvinyl, to kill the plant cells. So if a plant cell can produce a protein, or generally they're actually flavonoids that can kind of bind to the toxin and allow the plant cell to excrete the toxin or neutralize it, well, that's a win, right? That's one thing it can do without an immune system, develop a flavanol that binds to the toxin. Other things that they can do is thicken up their cell walls. Remember that fungi have mycelium, and they have to kind of go through the spaces in a plant cell wall. Plants can kind of thicken their cell walls or make physical barriers that the fungal mycelium cannot penetrate through. So there's a number of things that they can deal with, or plants have, even without an immune system. Now, before I go into a bit of a discussion here, we got to realize that there's kind of basically three types of fungi, or three ways that fungi are going to try to infect plants. There's necrotrophic fungi. These are diseases that kill the host cells and feed on dead tissue. So that's one. There's biotrophic fungi that feed on living cells, and they want to keep the cells alive for as long as possible. So something like tar spot is a biotrophic fungi. So tar spot is going to try to keep those cells alive. They're not going to try to kill them right away. Now, Fusarium is interesting. Fusarium switches from biotrophy to necrotrophy 2 days after infection. So this is a strategy or something called hemibiotroph. So Fusarium graminearum is a hemibiotroph. It's biotrophic first and necrotrophic second. Now, Fusarium head blight causes, Fusarium head blight basically enters the spikes, what you can see here, and it kills the spikes. So you can see that, it is, at the end of the day, necrotrophic. We see two varieties of wheat, AAC tenacious, which was considered resistant or is considered resistant. So you can see it's not fully resistant, but it is more resistant than Wilkin. When you look at Wilkin has a lot more damage. And when you look at the data here in terms of both infected spikes and the percentage of FDK, which is Fusarium damaged kernels, it's a lot higher in Wilkin. So Wilkin is a lot more susceptible than AAC tenacious. And the question is why? They actually figured it out. Now, one thing how do I put this? Yeah. One thing I got to say is if you're a plant and you're attacked by biotrophic, so let's say you're a plant and you're attacked by a biotrophic fungi. What is a good response? You don't have an immune system. Now, these fungi, you see that they want to keep the cells alive as long as possible. So one immune response that some plants do have to being attacked by biotrophic fungi is to kill their own plant cells. Basically, if they see the fungi advancing, the plant is going to kill its own cells, and that will essentially kind of stop the fungi dead in its tracks. This biotrophic fungi, if it wants to keep infecting plant cells, but all the plant cells around it are already dead because the plant killed them, then biotrophic fungi is kind of done. It has nowhere else to go. So that's a way plants can manage biotrophic fungi attack. But remember that Fusarium switches from biotrophy to necrotrophy 2 days after infection. It's a hemibiotroph. What this means is that if you're a plant and you decide to kill off your own plant cells because you see you're being attacked by a biotrophic fungi, that is playing right into Fusarium's hands because Fusarium is going to turn from biotrophy to necrotrophy. So it turns out that the reason, go back here, the reason tenacious is more resistant to Fusarium than Wilkin is that its response, its programmed plant cell death response, is lower than in Wilkin. Wilkin, which is the more susceptible variety, plays right into Fusarium's hands. Once it senses attacked by this biotroph, it's going to start killing its plant cells in advance to kind of block that Fusarium from continuing to move through the plant. But after 2 days, when Fusarium turns into a necrotroph, well, then it's still happy, right? Because it's going to feed on those dead plant cells. AC tenacious, in the more resistant variety, does not have as big of a response to the biotrophic phase. It will not kill off its own cells initially in the first 2 days when it's being attacked by a biotroph. There's some other mechanisms that it has, like it has ways to deal with the Dawn toxin. And so there's other things that drive resistance, but what makes AAC tenacious resistant and Wilkin not resistant to Fusarium head blight is Wilkin has a much higher response in terms of programmed cell death during the first 2 days after infection, when it thinks it's doing the right thing but playing right into Fusarium's hands. Our second and last case study is going to look at flavonoids. So flavonoids or flavanols are a class of molecules that play lots of different roles in the plant. So they play signaling, growth regulation, antioxidants, etc. Tannins, right, are flavonoids. So a lot of things that give plants or things we eat their flavor comes from flavonoids, different flavonoids. The thing that unites them all, there's thousands of different ones, they're all derived from the amino acid phenylalanine. Now, some flavonols have antimicrobial properties. And what that means is that flavonols can potentially protect against bacterial diseases if that plant can produce those flavonols in response, right? Plants would be wasting energy, right, if they were always producing lots and lots of flavonoids with antimicrobial compounds, because if there's no infection, that plant is just basically wasting like phenylalanine and other resources to do that. So this is a case study. We're not going to read the paper, of course, but it's a case study paper research done at the University of Guelph by Professor Gail Bozzo, Louis Lukens, others, looking at why the mechanisms why certain dry bean cultivars are resistant to common bacterial blight compared to susceptible ones. So dry bean breeders have breeded for resistance to or tolerance to common bacterial blight, but they did that without knowing like the underlying mechanisms, right? As a breeder, you're just growing plots in lots of different environments and looking at what's going to survive when there's a year with a lot of common bacterial blight. So they didn't understand the mechanism. So here you see a photo. What they did is in the greenhouse, they grew a susceptible and non-susceptible cultivar of dry bean and infected it with common bacterial blight. So you can see here's a susceptible leaf, or a leaf from a susceptible variety and non-susceptible. They call this resistant, but this is not 100% resistance, but it's just doing a lot better than the susceptible dry bean cultivar. So what's the mechanism of this resistance in the dry bean cultivar that really wasn't affected by common bacterial blight? Well, what they did is they took lots of leaf samples and analyzed them. This is a lot harder than it seems. Like you're taking a leaf sample and then looking for one molecule, one type of flavonoid out of many, many different thousands of flavonoids. So you have to isolate multiple flavonoids and test for them. It's complicated. What did they find? They found that the concentration of isoflavones, daidzein and gynstein, and related metabolites, coumestrol and phaseolinflavan, were increased in CBB-resistant recombinant and breadline plant leaves. So basically, these are all, like, I can't pronounce what these are, but they're all flavonoids. And the concentrations of these flavonoids was increased a lot in the resistant dry beans. Now, what they look is isoflavone pathway transcripts and metabolite profiles were unaffected in the CBB susceptible RIL. So in the CBB susceptible dry beans, these flavonoids were not upregulated. Okay, and so thus, what do they conclude? This is the conclusion in the paper. The induction of the isoflavone pathway is associated with CBB resistance in dry bean. Bottom line is that it looks like resistant varieties of dry bean upregulate the production of these flavonoids in response to bacterial infection, while the susceptibles do not.